Hi everyone, my name is Natalie Morgan. I am the Oncology Pharmacy resident here at Monument Health, and my clinical pearl this month is looking at carboplatin dosing. And as with most of my clinical pearls, this was a topic I wanted to understand better and understand some of the differences between our gynecologic oncology and medical oncology dosing recommendations for carboplatin. My objectives are to talk about a brief history of the dosing strategies that we have utilized for carboplatin, and then talk about some of the advantages and disadvantages and nuances and caveats associated with our famous Calvert equation for carboplatin, and then talk about some of the unique differences in terms of carboplatin dosing based on specific guidelines. So first off, talking about an overview of carboplatin in general. So carboplatin is a second generation broad spectrum platinum agent that was developed in 1981. And cisplatin, discovered in 1965, was kind of the precursor to carboplatin, although both are still widely used today. But cisplatin really did help usher in our modern era of chemotherapy. And both of these platinum agents are going to work by causing intra and interstrand adducts in our DNA. So they act very similar to an alkylating agent, but they do not have the addition of the alkyl group. But looking at that picture on the slide, there are some important structural differences between cisplatin and carboplatin, and particularly related to the leaving group. So our leaving group for cisplatin is going to be those chloride ions. In comparison with carboplatin is going to be that cyclobutane ring. And that ring actually adds a lot more stability to carboplatin and also increases its water solubility, which I thought was interesting to know. But in general, carboplatin is going to be less reactive and also be associated with less toxicity in comparison to cisplatin. So we see a lower risk for nephrotoxicity, ototoxicity, as well as emetogenicity with carboplatin. There is a dose-limiting toxicity, though, with carboplatin, and that falls into the myelosuppression category. And in particular, thrombocytopenia is going to be the dose-limiting toxicity with carboplatin. In terms of our dosing strategies with carboplatin, so we initially dosed carboplatin based off of body surface area using our milligram per meter squared like we do for a lot of chemotherapy agents. But with carboplatin in particular, when we used this dosing strategy, we found that it really did not correlate well with the person's glomerular filtration rate or their renal function. We also saw that there was a wide variation in the area under the curve, which is that patient's systemic exposure to the drug over time. And we saw really a variable risk for toxicity, and in particular thrombocytopenia. So patients would receive a lower dose, say 200 milligrams per meter squared of carboplatin, and have very severe thrombocytopenia. And then in contrast, patients would receive much higher doses, like 500 milligrams per meter squared, and have no toxicity. So it was just very difficult to predict. So our current dosing strategy does utilize area under the curve, so still focused on that systemic exposure to carboplatin over time. But now we've incorporated our famous Calvert equation that I'll talk more about coming up. But this equation has really allowed us to have a better correlation with the patient's renal function and their GFR. So you can see R squared of 0.886, but also looking at how that AUC value then also correlates with risk for clinical toxicity and in particular thrombocytopenia. And the Calvert equation has really allowed us to better predict both toxicity as well as efficacy with carboplatin. Wanted to touch briefly on the pharmacokinetics of carboplatin. So in terms of absorption and distribution, so it is mostly a hydrophilic molecule, has a relatively low volume of distribution, about 16 liters. In terms of protein binding, the actual carboplatin ring structure or structure in general is going to have no protein binding, but with that leaving group, then the platinum is going to irreversibly bind to our plasma proteins. It has minimal hepatic metabolism, and then half-life elimination for carboplatin ranges from 2.6 to 5.9 hours, but the action of the platinum and the binding there is going to persist for 5 or more days. And then for excretion, it's primarily going to be excreted in the urine, about 75%, predominantly with glomerular filtration. There is a small amount of tubular secretion as well, but important just to think about in terms of patients with poor renal function, lower GFR. They're going to have longer systemic exposure to this drug and increased risk for toxicity. On the other hand, patients with higher renal function, better renal function, while they're going to clear the drug faster and be at lower risk for toxicity, may also be at risk for not getting the full efficacy from carboplatin. So nuances there. And then in terms of the remainder of the drug, the about 25% remainder, that is going to be inactivated by binding to tissue proteins. Next, I want to talk about more specifically, our dosing strategies for carboplatin that we currently use. So first off, of course, I have to talk about the Calvert equation. And this equation, again, was due to this variability that we saw between, the patient's dose and then their risk for toxicity. So the Calvert equation was developed as a way to kind of help counteract that. It does adjust for the dose based on renal function. You can see the GFR in the equation there. And we saw that we could better predict the risk for thrombocytopenia and other myelosuppression. But there are some important caveats to know about the Calvert equation. So first off, with the GFR, in the original equation, that was measured using this radiopharmaceutical method, and it was allowed a very precise measurement of creatinine using this ethylene diamine tetraacetic acid, or EDTA, clearance. As you can imagine, in clinical practice with a radiopharmaceutical, that is not a very easy measurement to obtain. So now we have utilized more formulas that help us estimate the creatinine clearance and estimate the GFR, which does in some ways limit the accuracy of the Calvert equation. Most commonly, they're going to be using the Cockcroft-Gault or Joliffe equations for estimating, and I'll talk more about those coming up. There are also some general controversies with the Calvert equation, and of course all of these factors really are concerned about variables in dosing of carboplatin and what dose the patient actually receives. So there have been, as we talked about, various equations to estimate the GFR because not easy to obtain, a radiopharmaceutical and a precise EDTA measurement of GFR in clinical practice. But these equations, particularly Cockcroft-Gault, is going to be dependent on the patient's weight and serum creatinine, which does increase variability in that estimation. There's also been discussion about capping the GFR or allowing a maximum GFR of 125 mils per minute because that has historically been the maximum that the kidneys have been thought to be able to filter. And then also serum creatinine as a measurement of byproduct of muscle metabolism. Oftentimes for patients that have low muscle mass, they may have a falsely low serum creatinine that may not actually reflect their true renal function. So there has been consideration for using a minimum serum creatinine of 0.7. And then dosing in obesity is also a controversy, and in particular, what body weight do we use in our equation? So looking at ideal versus adjusted versus actual body weight, and looking at our dosing, looking at body surface area versus AUC versus Calvert equation. Important to note that there have been no head-to-head studies looking at the differences in these dosing strategies, but we do have data within each dosing strategy, which is important to note. So specifically looking at carboplatin dosing in obesity. So for definitions, overweight patients are going to have a BMI between 25 to 30, and then obese patients have a BMI greater than or equal to 30. But the body weight in our Cockcroft-Gault equation is meant to reflect muscle mass. So that's important to think about for our patients with obesity. Also, adipose tissue in general is going to have a lower proportion of water than muscle tissue, and with carboplatin being a hydrophilic molecule, it's not thought that it's going to distribute very well into the adipose tissue. And then there is a risk for, in patients with obesity, using the actual body weight in that equation can actually overestimate their creatinine clearance. And then that would give us a higher than anticipated carboplatin AUC and risk for overdosing. So again, using that adjusted body weight may actually better predict carboplatin clearance. Next, I want to talk more specifically about the equations that we have for estimating renal function. So for creatinine clearance, GFR is actually, the gold standard for measuring GFR is inulin. And inulin is used because it is completely filtered at the glomeruli. However, just like we saw with the EDTA measurement, this is not easy to obtain in clinical practice because it requires a continuous IV infusion of inulin over a prolonged period of time. And then we have these methods for estimating GFR that are reliable in a lot of circumstances, but in other populations, they may not actually reflect the true renal function. So like we talked about, patients with low muscle mass have a low overall production of creatinine, but that may not actually reflect their true renal function. Also, similarly for our elderly patients that have lower muscle mass. So there can be issues there. In general, though, we do have methods where the creatinine clearance can very closely approximate the GFR. So in particular, if we obtain a 24-hour urine and measure the patient's serum creatinine levels, that can be very accurate. As we talked about, the EDTA method that was used in the original Calvert equation, looking at a radiopharmaceutical as well. So these are both, accurate methods that we have, but just difficult to obtain in clinical practice. And then this was a new finding for me, but actual lab measurements of serum creatinine have changed relatively recently. So in 2010, there was a standardization that occurred in all US labs where they were required to have their serum creatinine values calibrated to this traceable isotope dilution mass spectrometry, or IDMS reference. And this was actually found with this standardization. The hope was that we would have better standardization across all of our labs in the United States, but this was actually found to give a lower estimate of the serum creatinine. So then in turn, it led to a higher GFR for that patient, higher estimated renal function. And there was a risk for then giving that patient a higher dose of carboplatin. So with this standardization, the FDA actually released guidance that there should be a maximum GFR of 125 mils per minute when calculating carboplatin doses, which then gives us a maximum dose per designated AUC value. And this is just a picture of the FDA statement related to or in response to this standardization that came out in 2010, again saying that this IDMS calibration may actually underestimate the creatinine, give us a falsely low creatinine, and then we would have estimation for a better renal function and risk for increased carboplatin dose, which I found very interesting. And this is just a study I found looking at the differences between pre-standardization and then after the IDMS standardization. So on the y-axis, we have the carboplatin dose in milligrams. And then on the x-axis, we have 5 different sort of patient generalizations that are represented. And then in terms of our black and white bars, the white bars represent the carboplatin dose prior to the IDMS standardization, and then the black bars represent after that standardization in 2010. So you certainly can see that we are giving higher carboplatin doses with that standardization in 2010. But then also interesting to see some of the differences in terms of patient generalizations. All right, so looking more specifically at 2 equations that we use for estimating creatinine clearance. So first off, we have the tried and true Cockcroft-Gault equation that we're all familiar with. So this was developed in 1976, and it was based off of a study of 249 Caucasian males. And they did include a wide age range. They were aged 18 to 92. They also included variable renal function, so patients with and without chronic kidney disease. But they're important to note that there were no female participants in this study, so the coefficient that we use for female gender of 0.85 is hypothetical. And then also with our body weight, the Cockcroft-Gault originally was not built to account for adjusted body weight, and important to know, especially as we see an increasing incidence of obesity in our population. But looking at the numerator, certainly, that age coefficient is large for the Cockcroft-Gault and can lead to an underestimation of creatinine clearance in our elderly patients. And then, as we mentioned, the body weight and not being designed for adjusted body weight for patients with obesity. And then our serum creatinine, as we talked about with that IDMS standardization, can lead to an overestimation of the patient's renal function. And then we have our Joliffe equation, and this is a formula that was developed in 2002, and it was actually developed to estimate renal function for patients that have rapidly changing renal function and are acutely ill. So the big benefit here is that there's no factor for the patient's body weight, and so this equation was actually largely used in our trials for gynecologic oncology malignancies, and up until really the standardization of the serum creatinine was used commonly, but has now fallen out of favor, and I'll talk more about that coming up. So with that in mind, that brings me to talking about our guideline recommendations for carboplatin dosing. So first off, I want to talk about our Gynecologic Oncology Group, or GOG guidelines. So as we talked about, the Joliffe equation was widely used in our GOG trials to estimate creatinine clearance. And then there was a trial, the GOG 158 trial, that looked at obese patients and actually found that they were at risk for underdosing of carboplatin. And so in this trial, they used the Joliffe equation, which again does not account for body weight. And when they stratified by patients who were obese, they found that these patients had less toxicity, but also lower survival. So especially for patients with a BMI greater than or equal to 30. And then also, as we talked about with the standardization of the serum creatinine measurement with the IDMS calibration in 2010 and the FDA warning about an risk for increased carboplatin toxicity, this led to a shift in the GOG recommendations. So now they recommend use of the Cockcroft-Gault for creatinine clearance estimation, using a minimum serum creatinine of 0.7 and a maximum creatinine clearance of 125, as well as an adjusted body weight for patients that fall into that overweight or obese category, so starting at a BMI of greater than or equal to 25. Our NCCN guidelines are very similar to our GOG guidelines. They're going to recommend using the Cockcroft-Gault as well for our estimation of GFR, and then using a maximum GFR of 125 mils per minute. They are not as stringent as GOG in terms of serum creatinine and body weight, though. So they recommend consideration for a minimum serum creatinine of 0.7, especially in our elderly patients or those with cachexia. And then they also recommend considering obtaining a 24-hour urine or an EDTA as a radiopharmaceutical measurement to estimate creatinine clearance especially when you're using high doses of carboplatin, so AUC above 6, or if there's an uncapped creatinine clearance. And then for our overweight or obese patients, again, they recommend considering using an adjusted body weight. Similar to GOG, they also have maximum allowable doses per target AUC. And then last, we have the Alliance for Clinical Trials in Oncology, and this was a newer organization for me, but does have some important differences in comparison to GOG versus and NCCN. So they also recommend use of the Cockcroft-Gault to estimate GFR and using a maximum GFR of 125 mils per minute. But they recommend using an actual serum creatinine as the lab reported value. Again, they recommend considering obtaining a 24-hour urine if there's a concern that we're not obtaining an accurate creatinine clearance. And then for overweight or obese patients, in contrast to NCCN or GOG, they recommend using an actual body weight. And I just pulled some of the wording from a clinical trial protocol with Alliance. And so you can see they state failure to use actual body weight will be considered a major protocol deviation. So important difference there. And then they also have a maximum allowable dose per target AUC, just like NCCN and GOG. So that brings me then to our summary of carboplatin and carboplatin dosing. So first off, going back to our history of carboplatin, this is a second-generation platinum agent that was developed in 1981, and it is generally a less toxic version of cisplatin, although both agents are still widely used, but we do see lower risk for nephrotoxicity, ototoxicity, and emetogenicity with carboplatin. And both of these platinum agents are going to work similar to our alkylating agents by causing intra and inter-strand adducts with our DNA. But carboplatin, we're really going to see the dose-limiting toxicity being myelosuppression, and in particular thrombocytopenia. Carboplatin was initially dosed like we dose a lot of chemotherapy using BSA, so milligram per meter squared, but this was found to actually correlate poorly with AUC as well as toxicity in clinical practice. And so we have this unique, famous equation, the Calvert formula, that has helped us to correlate AUC with both efficacy as well as toxicity with carboplatin, so a very unique formula. So looking in particular at our Calvert equation and some of the important features to know about this equation. So looking at the GFR, this was originally measured using a precise EDTA clearance, so the radiopharmaceutical method. Unfortunately, this is not easily obtained in clinical practice. So we have these imperfect measurements for estimating GFR and creatinine clearance as a substitute in our Calvert equation. We have the tried and true Cockcroft-Gault equation. It's going to have variables with both serum creatinine and body weight that can influence the estimation. And then we have the Joliffe equation, which was commonly used in our gynecologic oncology trials up until recently because it does not rely on body weight. However, with the 2010 update to our serum creatinine and our standardization across all labs in the US with our IDMS reference, the FDA released a warning that there's a risk for underestimation of serum creatinine with this calibration, and that this can lead in turn to falsely elevated creatinine clearance levels and then a risk for overdosing. So based on that, all of our main guidelines are going to recommend using the Cockcroft-Gault for estimation, and the Joliffe has fallen out of favor. Last, I just wanted to provide an overview of our three main guidelines, so NCCN, GOG, and Alliance. And so all three of them are going to recommend a maximum GFR of 125 mils per minute for our estimation. And then NCCN and GOG are going to recommend considering a minimum serum creatinine of 0.7, especially for our elderly patients or those with low muscle mass. In contrast, Alliance is going to recommend using the actual lab-reported serum creatinine. And then for our patients who are overweight or obese, for NCCN and GOG, they're going to recommend using an adjusted body weight, even though, as we talked about the Cockcroft-Gault, that was not the body weight that it was designed for. In contrast, Alliance is going to recommend using the actual body weight regardless of if the patient is obese or overweight. So I hope that this has been helpful in terms of understanding some of the differences and nuances with carboplatin dosing strategies and how we came to develop the Calvert formula and other important details in terms of the guideline differences. Thank you so much for listening to my clinical pearl.